Okay, so today Dr. David Rothline from Boston University will be speaking with us. He's a postdoctoral research fellow working with Dr. Michael Isterman, and he'll be talking about mapping the fidelity and connectivity of visual information using fMRI, implications for attentional control, valence processing, and PTSD. Please help me give a warm welcome to David.
Hello, can everyone hear me okay?
Yes. Yes.
Yeah, good. Okay. Um, Thank you very much for the introduction. Uh, Basically, so today I'm going to talk about, as the title said, two projects that I'm currently working on that are aimed at sort of getting at the fidelity of information in the brain and the connectivity of this information and how it relates to valence processing, attentional control, and PTSD. Um, so yeah, basically struggling to come up with a way to unify everything, but I think the thing that they both share is it's trying to get at a very precise measurement from the bold signal. So to sort of set this up, I want you to imagine that you're in an MRI scanner, which this is a picture of the scan, an old picture of the scanner at the VA hospital in Boston. And imagine you're in the scanner and you're looking around and you see, you're looking up because the person running the experiment tells you you're going to see images and you see pictures of objects like the horn and telephone, and then you see pictures of faces. And then we're measuring your responses and we see some pattern like this. So when you're looking at faces in these brain areas, the area highlighted in green, which is the fusiform face area, you get a pretty good signal. And then when you are looking at objects, the signal goes down considerably. And so we have this result right here, and there are sort of, particularly with fMRI, but with many different types of measurements, there's different kinds of ambiguity that can arise from interpreting a result like this. So one is basically, how well does the thing you're measuring actually relate to the thing you want it to measure? So in this case, the question would be, how well does the bold signal in fMRI measure what's actually going on in the brain? And so I don't know how much background everyone has, but basically the way fMRI works is that it's a fairly indirect measure of neural activity. It's sort of trying to get at, some response to a lot of metabolism that's going on in a particular brain area. You get this signal of oxygenated blood coming rushing back in, and you see that signal in the MRI. So that is one of the measurements, and so there could be some ambiguity there, but I think a lot has been written about this level of ambiguity, and I think the next is sort of uh, more spreading across a lot of the issues when it comes to psychology and cognitive science in general, which is how well does the bold signal in this case reflect the specific mental process that you're trying to get at. So we have this experiment where we're looking at faces and objects, and we want to know, are we really tapping into something about an object being a face, or is it just, round things, or is it just, things, anything with hair would activate this, or any sort of visual object that falls within the same category, you'll sort of activate this area over time. And so it's sort of ambiguity in this sense that's not limited to fMRI, but it's certainly very prevalent in fMRI. And so that's sort of the challenge that I think I want to try to chip away at a little bit. And so in the case in the past with univariate designs like this, what people did, and so this is from a seminal paper by Canwisher, where they identified the fusiform face area as being a region of the brain that sort of really likes faces. They didn't just show faces versus objects. They had to, try to rule out other options, like maybe let's see if it still responds to scrambled versus intact faces. So we can rule out like subface features, or maybe houses. Do we still get the same category? Or maybe if we look at faces that are rotated and compare it to hands, and maybe if we change the task, do we still get the effect? So really, that paper wasn't just looking at faces and houses. They did a lot of different experiments to sort of get at whether, this is the face area, but of course, the number of things you could possibly compare it to is almost infinite. So really, it's hard to sort of narrow down with a bold signal, precisely what that signal is reflecting, particularly if we don't have a really good cognitive theory to sort of say, these are the things I'm looking for. So that's sort of the ambiguity in fMRI-based measures, which again, also true with reaction times or EEG or any kind of measure that you could have. I'm going to sort of set myself up for failure because I don't know how car engines work, but I do really like this analogy that I heard, which is that using fMRI and bold signal and activation to infer cognitive processes is kind of like trying to figure out how a car engine works by looking at what areas get hot when you turn the car engine on. Now, again, I don't think those are the areas that would get hot. I think most of the engine would probably get hot, but you can see that's certainly an effect of you turning the engine on and the engine starting to work. And there will be a distribution of where the heat is on the engine, and there will be correlations among those, but in the same way that, we often correlate brain activity across brain regions to get at, are they communicating? But in this case, it's not that they're communicating. It's just that, when the engine heats up, other things start moving, friction starts happening, and you get heat going a lot of different places. And so again, it's challenging to sort of make that jump. And I don't claim to have solved it here, but I do think I have some ways of at least getting a little bit more clarity. And so I'm going to talk about two studies. The first is one where I use representational similarity analysis in a sample of veterans to sort of try to assess the fidelity of the visual information that they're seeing during a sustained attention task. And also to see if we can see how the visual information, if not is being transferred from one brain region to another, at least is sort of concordant or consistent across brain regions. The second study, I'm going to use a different method, not representational similarity analysis, but inter-subject synchrony, which is often used in movie watching paradigms or naturalistic stimuli paradigms. And I'm going to also show you sort of how we manipulated the order of a trial orders across different runs in order to try to get at see if we can disentangle simultaneous overlapping streams of visual information. And so the first approach in the first study is going to use representational similarity analysis. And so one of the main things that representational similarity analysis is trying to address. I should say, please stop me at any time. I don't know if I can see if somebody's raising their hand, but you can just make a noise to interrupt, or and because again, if I'm moving too fast, I could definitely slow down and explain things a little bit better. So the problem that representational similarity analysis is trying to address is that oftentimes when we're looking at a particular measurement, we don't really know how that information is encoded. So it's sort of a more subtle way to say the exact same thing. We don't know exactly what we're looking at when we see these signals. And so with fMRI, it's generally not a problem for broad categories because you can sort of see, big visual differences in the brain activation maps between, for example, faces and scenes. And so in a way, when you're looking at that brain map, you're sort of, doing multivoxel pattern analysis yourself by saying, I can see this is probably looking at faces and scenes or something like that. So you can see from looking at a brain map, kind of, if you've looked at enough of them, you can kind of tell what the task probably is. However, if we were to just compare, within a category, when you get into subtler details or subtler differences within sort of a category or subcategory of objects, then it becomes a lot harder to, determine what kinds of processing we're getting at. But also, I can definitely, for example, if we look at these scenes, I can definitely distinguish the, country road from the farmland, I guess the forest road from the mountain. Like those are very visually easy for me. But if I were to present them in a scanner and look at the brain results, I don't think I could tell which one you were looking at. However, that information is certainly encoded in the brain. And so the challenge here is to try to get at more subtle ways of analyzing the data to see if we can get, the kind of granularity of, what differentiates looking at, within these cellular differences within a category. And so again, we're looking at across multiple voxels. You can imagine just stretching them out. But if we take a brain region and we get an activation pattern, you can measure one activation pattern for the forest road, one activation for the farmland, and another activation pattern for the mountain. So we get different vectors. Basically, you can just think of these vectors of numbers. And what you can do is you could say, okay, well, how similar are they to one another? And so this is sort of the, oh, sorry, going the wrong way. This is sort of the approach between of representational similarity analysis. So you could say, how similar is the forest road to the farmland? And you can do a correlation of these activation vectors to get a similarity value. And how similar is the forest road to the mountain? And you get another correlation value and so on. And you can sort of fill out this matrix, which would be like a covariance matrix, but it will also remove the stimuli from the encoding of a particular ROI and put it into this common similarity space. And so in this similarity space, you can do a lot more with this information because you don't have to know what each voxel is doing in this activation pattern. For example, you've taken it out of there and now you can test it against, my own similarity judgments or some computational model of how physically similar these images are, their pixel properties, and so on. And so, and yeah, and so basically, uh, the point of representational similarity analysis is sort of taking information out of these different encoding or different ways of encoding the data. So fMRI, computational models, cell recordings, brain regions, across people, across brain regions, and putting them all into this common similarity space where they can be compared directly to one another. And so that is, I think, the real nice thing about representational similarity analysis. It allows you to make comparisons that would otherwise be a lot harder or a lot more computationally intense. And to me, it's just more intuitive to think about it this way anyway, instead of trying to fit a complicated model to one set of voxels to another. And so the task we're going to be using to sort of use this method to get a better understanding of the visual information is something called the gradCPT, which is a sustained attention task where basically you have to press a button. You see it's gradually fading from one to next, and you press a button every time you see a city scene. And then when it's a mountain scene, you withhold from pressing. So you can sort of see press, 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 press. Is there a mountain? Yeah, there we go. Withhold, press, press, press. And so you see it's pretty challenging. They move pretty fast. And most of the time you're pressing buttons, and occasionally you won't press a button. And so this is the task that participants are doing in the scanner. And this is the kind of stimuli that we want to try to get visual information from. And so basically they're doing it for a while. It's a well-validated experimental paradigm, and we can track fluctuations in attention, but also we can measure overall measures of how accurate someone is, or how many errors they make, and whether they press or accidentally withhold from pressing when they're supposed to. And so we have this task, the gradCPT, and there's importantly 10 city exemplars. And so because the stimuli move so fast, we can actually, each city exemplar is repeated on average 54 times a run. And so this is nice in fMRI because even though the stimuli are presented, way too fast for normally for event-related fMRI, because they're repeated so often at random and the order is randomized, we can actually estimate activation patterns using the standard, way with fMRI with running a GLM. We can basically model, okay, how does the brain signal look every time you look at city exemplar 1? How does the brain look what is the pattern, or how does the brain look every time you see city exemplar 2? And we can estimate these activation patterns for each of the city exemplars. And again, they're pretty similar. There's nothing too strikingly different. So this isn't a case of like cities versus faces. These are all going to be pretty subtle differences that you wouldn't be able to tell by eye, but you could, make judgments about how similar these city scenes are to one another. And so there is definitely a structure there. And so we're able to estimate these patterns of activation for each. And then what we'll do is, again, this is an example of looking in the PPA. We can then correlate these patterns to see what the similarity structure is within the parahippocampal place areas. So we have our vector of activation patterns from the GLM, and then we cross-correlate all of the possible exemplars, and then we form, so there's going to be a lot of steps here. I thought I broke it down a little bit more. Sorry that I didn't, but I'll try to go through them one by one, even though it's all there. So basically what we can do is from these cross correlations for each participant, we'll form an activation or a representational similarity matrix like I showed before. So this is again, is just all the cross correlation values, the Pearson R values of each of the city's exemplars patterns within a given brain region. So we'll call it ROI1 here. And so what then a really nice thing, and this is how we sort of assess the fidelity and the connectivity in this case, is that a lot of participants did the task. So we had actually 140 veterans take this task. We have an independent sample of about 80 people doing many runs across many sessions of this task. And so we can take, for example, the people who did the 80 runs, the non-veteran sample, and we can look at how their activation is or how their similarity structures are across many different brain regions. So we can look within an ROI, we can estimate each participant's representational similarity matrix, and then we can actually take the group average of all of them, because again, they're all in similarity space, so they can be compared directly, they can be averaged. And presumably if the noise here is not too skewed and it's symmetric around the mean, what happens is when you average them together, the noise is going to cancel out and you're actually going to get a better estimate of what the actual similarity structure is, if there is one. If there's no signal there, then they're going to cancel out and you're just going to be left with noise. But if there is a signal, you average together, you're sort of estimating what the similarity structure is, even if you don't have some kind of prediction in mind of what it should be. And so here we take one sample, we estimate what the similarity structure should look like by averaging the whole group together. And then we have the sample of veterans or the sample we're interested in, and we can measure the fidelity by saying, okay, how well does this given participant's similarity matrix within this brain region correlate with the group average with our best estimate? So it's basically like saying, okay, how well does when I see these scenes, how well does the similarity structure in my visual cortex relate to your visual cortex? And we can see if there's anything, since we're all seeing the same stimuli, if there's anything shared, then they should correlate. If there's no shared information, then it's just going to be noise. But the nice thing about doing it this way as well is additionally to looking within a brain region, we can also do it across brain regions. So we can say, how similar is my visual similarity structure for these city scenes in my visual cortex to your parietal lobe or dorsal attention network, broadly speaking. We can sort of compare across regions as well and to see are they, seeming to at least organize the information in the same way. And so again, what I'm hoping to get at here is the fidelity in this case, because we're doing it across participants, or it's in a cross-validated way, and because it's sort of looking at just the fine-grained details of the city scenes, whatever fidelity comes out has to be something about the city scenes that's organizing them or differentiating them at a pretty narrow level. So we can say that signal is pretty clearly about the visual information you're looking at. And not about, some sort of attention, general processing, or task modulation, or something that is just abstracting the cities and mountains. You're looking at a signal that relates to the visual information. And so when I do a searchlight across the whole brain, what you find, which is always promising, is that you see the signal is, where you might expect it to be for visual information, which is in visual cortices and sort of extending up along the dorsal stream and along the ventral stream and into maybe a little bit of the parietal lobe. So that's where you get the overall signal. Now, I didn't really set up the motivation here, but this is a sample of veterans. And so, well, I'll first say we've replicated this finding basically across many different data sets as well. And you'll see, there's some differences, but by and large, you get a strongest signal in visual cortex, and then you get to some degree it extending up along the dorsal attention networks into executive networks and stuff like that. Um, so, uh, so there's a couple of questions now. Since we established that we have the fidelity for each participant, we can now ask, okay, well, does this measure of how, well the participants, or how consistent at least the participants are measuring or are representing this visual information, does this tell us anything about how they did on the task? And does this tell us anything about how they just process visual information more generally? So first we want to say, does it have some sort of is it sensitive to how they performed on the task? So you might expect that if you have better fidelity, if you're representing the information more cleanly, or at least more consistently with what everyone else is doing on average, then you might expect that you're paying more attention, you're looking at the task, you're not looking away. So you might expect that you're doing better in the task. And in fact, that's exactly what we find with this data set. So in visual cortices specifically, the better your fidelity is, the you have a correlation with the accuracy on the task. Um, uh, Get that out of the way. I don't know. Okay. Okay. So yeah, basically again, and this is true actually across multiple data sets as well, that you find that your accuracy on the task is correlated with visual, fidelity. So how clean it is in the visual cortex. Now, also what I think is really interesting here is we ran this in a sample of veterans who have varying degrees of PTSD symptomatology. And so one of the possibilities is that there's different ways of processing information that's pretty neutral. And so, and it's in fact not the emotional, negative images and positive images that necessarily distinguish how, your mindset, or how something like PTSD is hypervigilance, they're looking for threats. It's not necessarily in the situations where you should, have a normal threat response if something is upsetting. It's when it's neutral and the stimuli aren't necessarily very threatening, but you're sort of over generalizing or over applying this. And so we were interested in seeing, okay, with these city scenes that are fairly neutral, is there any relationship of this fidelity, how much they're processing it and where they're processing it, more importantly, to their CAP score, which is basically a measure of PTSD symptomatology. And so what we find, interestingly enough, is that there are differences in the fidelity between, where they're processing the information and whether it is predictive of less PTSD symptoms or greater PTSD symptoms, specifically within the amygdala and this sort of insular region on the left and right side. Actually, you have an association with higher CAP score, higher symptomatology. Whereas if you have this more dorsal network processing of visual to dorsal attention network, you actually have lower symptoms. And so it's sort of preventative. So it seems like we're setting up for like two different streams of processing here, where if you're processing the scenes through one route, it's predictive of having, a higher CAP score. And if you're processing it through a different route, it's predictive of having a lower CAP score. And so we took a parcellation approach and decided to look at the connectivity instead of just the fidelity. So now, instead of correlating the similarity matrices within a region for the fidelity, we're now going to do it across regions. And so what happens when we look at like the visual, my visual, similarity matrix to your parietal frontal or temporal frontal, et cetera. And we can get these connectivity values and see, do they relate to your CAPS scores? And we find, well, first, errors on task, consistent with what we showed before. If you have more connectivity between the parietal frontal and the temporal frontal networks, you have fewer errors on the task and a higher D prime. But more importantly, looking at the CAPS, we find that if you have more connectivity between the visual and these cortical regions, then you actually have a lower CAP score. So it seems to be preventative, or at least it's not clear which way the direction of causality is. It's not if it could be that having this sort of mode of processing is protective. It could be that having PTSD causes you to use this pathway less. That's less clear, but at least at this point, we can say that it's associated. But importantly, we find that when you have visual to amygdala connectivity, you have higher CAP scores. And what I think is the most interesting is when we look at amygdala connectivity, the cortical networks, we find that it goes in the opposite direction. So connectivity from the amygdala to all of these cortical networks is associated with greater CAP scores. So the reason why I think this is cool is it suggests that the amygdala is sort of like not hijacking, but routing the information, sending it to the same brain regions to make decisions on it, but coloring it in a certain way or viewing it through a certain lens, or maybe adding some contribution as to how it, how one is now expected to respond to this information. So I think there's a lot of interesting questions here to say what this means, but what it suggests to me is that there are these sort of two pathways of processing these neutral visual information, and it's the neutral information that's sort of telling us, is this really about, is this sort of, we're maybe overgeneralizing threat or overgeneralizing some sort of hypersensitivity in this pathway, or is it more just that you're, yeah. Anyways, so basically that
David,
is
can I
the,
ask a
yep,
question real quick?
yep, definitely.
Sorry, just a clarifying question. Are you looking at task-based functional connectivity, or is it some type of like fidelity of connectivity measure like you were doing before?
This is this is representational connectivity. So this is it's based on the task, but instead of looking, whereas the representational fidelity was within a region, looking at how similar the similarity structures were within a particular brain region. The representational connectivity is looking across brain regions, so from ROI1 to ROI2. And so that's basically, when we look at these results here, what we're saying is basically, if the similarity structure in the visual cortex is similar to the visual similarity structure in the amygdala, then that participant is more likely to have CAP score. And if the visual similarity structure in the amygdala is similar to these cortical regions, then they're more likely to have higher CAP scores, whereas vice versa for visual straight to these cortical networks. Does that make sense?
Yeah, definitely.
Yep.
Thanks.
Yep. All right. So basically that's the first study where we use representational similarity analysis to try to get at this higher fidelity, or to get at measurement of fidelity of information. The second study is going to use intersubject synchrony. And so basically this is one that's very new and I'm excited about it. Again, apologize for the AI generated image, but I couldn't resist when I saw it. And so basically, the problem conceptually that we're dealing with is that in real life, you're bombarded by multiple streams of information. So it's rarely the case that unlike in a lab where you're told to focus on a single thing and you're told to ignore everything else, you're basically, instead of, focusing on a single thing, you're focusing on the thing and selecting what's goal relevant, but at the same time, you do want to monitor everything else in the background, because that might change your goal. So you might get a call, or you might hear a fire alarm go off or something, and so you want to not be completely engrossed in a task. You still want to be monitoring this information, but at the same time, you want to control. You don't want to be too focused on what's going on in the background as well. So you have to sort of calibrate this, because in this competition, in what you're paying attention to could lead to lapses in attention. And so, because this seems to be a very important part of what we do in the real world, the goal here is to use task and fMRI to sort of measure how we allocate these multiple simultaneous streams of information in the brain. And so the task we're using here is a sort of modification of the first task I showed you, and this is called the EmoGradCPT. And so just to set it up briefly, instead of seeing images of city and mountain scenes, we're going to be seeing digit or we're going to be seeing numbers basically. And instead of, pressing a button every time it's a city, you're basically going to press a button every time the number isn't a 3. And when it's a 3, you withhold from pressing. And then in the background, you'll see there's some pictures, and you're just supposed to ignore those pictures. So let's see if I can get that going. Yeah. So we press. Oh. And so you see you press whenever it's but then you withhold when it's a 3, press, and you try to ignore the cute images we're seeing right here. And yeah, so that's basically the task. And so one thing that you'll notice is that there's multiple things going on. You have the background that's pretty hard to ignore, but basically would tell you you have to try your best to ignore it and just focus on the numbers. So the question is how much of that background is getting through, even though we're telling you to ignore it. And the other component here is that the images are blocked. So I only showed you the pleasant one, but there's more upsetting images as well. So it's blocked in terms of positive images and negative images. Where negative, it's not terrible, but it's definitely not great. Roadkill, crying babies, stuff like that. And so one way to get at least behaviorally, is are these background images coming through? Is do we see differences in terms of the negative and positive images behaviorally? And in fact, we definitely see that when the background images are negative, you perform your D prime, your accuracy on the task is lower. And as opposed to when the objects are neutral and positive. Furthermore, your reaction times, I don't think I have it plotted here, but your reaction times are slower when the background images are negative. And so clearly, you're able to get some of this information is getting through, because even though you're supposed to ignore the background images, they're still influencing your performance. And it's also true that like more negative images are make you worse at the task. And so the goal here is to see if we can find a way to basically disentangle, because we're seeing these two simultaneous streams of information. And I go in the scanner, and let's say we have this task, and this is just a representation of, the digits you saw were much bigger than this, but basically you have 4 digits per background image, and they gradually fade from one to next. And so we do this in the scanner, we measure a signal in the brain, and this is the signal that we get. And so the question is, what part of this signal is going to be due to the digits, and what part of this signal is going to be due to the background? Because in order to make some kind of conclusion about what we're seeing in terms of what brain is doing, you want to know, particularly when two things are going on, how can we disentangle these signals? And so the method I've come up with here is actually using the trial order and intersubject synchrony. And so the idea here is that when participants see the exact same thing, so this is true of like movie watching, for example, you're able to basically say, look, I can't model everything that's going on when you're watching a movie. However, I can say, if the time course is locked to the same segments of the movie, I can average them together, and we can see what sort of a group average is of, an estimate, in the same way I did with the similarity matrices, estimate what the shared voxel response time course is. And then you could take each participant and say, how similar is that participant to this group average? In a similar vein, we can do something like that with this task. And even though it is in a movie, we can play with the trial order. So we can have each participant run 3 types of runs. One where they basically see this particular order, which is the puppy and the 2 babies, and then this particular sequence of digits. And every participant is going to see the exact same sequence. And so we can, in the same way you would do it with movies, where you can basically correlate across participants, you could in principle do it with this as well, but you still wouldn't know what part is the foreground or background. But then what we can do is we can run a second run where we try to estimate, okay, what about just the background or just the foreground signal? And so in this case, we'll fix the order of the foreground to this 7, 8, 1, 5, etc. digit sequence, but we'll randomize the background order. So one participant will see, these pictures. Another participant will see these pictures, but have the same exact digit sequence. And then we can begin to, average the signals together. And basically what we find is when you keep doing that, you can basically distill out or, pull out the activation or the time course related to just this digit sequence, and the backgrounds are kind of averaged together and wash out as being just some mishmash of, whatever. And so the hope is when you average these together, we're basically amplifying the response patterns due to the digits and sort of mitigating the responses due to the backgrounds. And then we can do the exact same thing with the third run, where we average together the digit or randomize the digits, but then keep the backgrounds in the same fixed order. So then we get a signal that is basically, the association of just this background and not the digits themselves or the particular digit identities or the task, dynamics. These will all wash away. And so finally, for each participant, then we go back to the first run, and we can now estimate, using these estimates, we can sort of pull out with a regression how much of this signal is due to the digits and how much of this signal is due to the backgrounds. And we sort of run this regression for each participant for each brain region we might be interested in. And we can then look at, how much of this signal, and when we do this, we see for the distractor scenes, even though you're supposed to ignore them, they're still very big scenes and they're much more visually prominent. We get a lot of signal throughout the whole brain. And in fact, coming into this, I wouldn't have been surprised if we only get these background images, because if doing this for a little while, visual stuff really comes out strongly in fMRI, and the less visually salient it is, the weaker the effects tend to be. However, when we look at the task digits, it's really interesting. You actually get very little within these lower level visual areas, probably due to the regression, but you get very little in these lower level visual areas, but you start to get a lot more signal actually in these higher level attentional and executive brain regions in the parietal and frontal and motor cortices. And so it seems to suggest that emerging from the background that you're supposed to be ignoring, you're sort of magnifying the digits and pulling out the information that you're supposed to be doing with the task. And so again, I basically making the exact same point. You can see that just when we look exclusively, the background scenes are much more prominent in lower visual areas, where the task digits in orange exclusively come out in these sort of higher level regions. But also importantly, there's plenty of regions that seem to be monitoring both. So these can sort of act as decision points, or basically areas that are like constantly weighing whether or not you should switch your attention or whether you shouldn't switch your attention. Okay, again, we can always get into this, but since we're running low, I can just sort of highlight a little bit, which is we could do the exact same thing and do the exact same method, but now just split it. Okay, what if we only look within the positive scenes, and what happens when we do this exact same analysis, only looking within the negative scenes? Do we find brain regions that, for example, might switch between the positive and negative in terms of how, like, what you might predict from the behavior? So looking at these, we can sort of find many different types of effects. So just to highlight what we're doing a little bit, we have this right dorsal attention network region that for the distractor scenes, that's what the DS stands for. This is the one in yellow, if you can't see my cursor, the one in bright yellow. Basically, what you find is that for the distractor scenes, when the scenes are positive, you have a little bit more activation, but when they're negative, you have less. So basically, oh, sorry, I had it backwards. Yeah. Sorry, I was saying it backwards. That's why it's confusing. For the distractor scenes, when they're negative, you have a little bit of activation going on. You have some synchrony going on across participants. So it seems to be representing the negative scenes, but when they're positive, it completely ignores them. However, for the digits, when the digits are overlapping negative images, it's far less synchrony than when they are overlapping positive images. So what you have here is a clear case where there's competition between the negative distractor scenes and the task digits. When the distractor scenes are negative, but when they're positive, there really isn't any competition. The task digits sort of lead. And so we're able to identify regions that seem to be responsible for this switching in terms of based on the emotional valence of what you're looking at. So yeah, the conclusions here is that it seems task relevant digits emerge from the visually dominant distractors. And also this design of fixing and randomizing the order enables us to look at how the background distractors interact with what you're supposed to be doing. And perhaps in the future, this can be used to identify individuals who struggle with their ability to ignore the backgrounds or are too eager to, look away or find things. We could also use it more generally to find what types of things in the world are the most distracting or what brain regions modulate these. So yeah, that's basically it. Thank you very much.
Thanks for a great talk, David. I'll leave it open for questions for a few minutes if anyone wants to come up. Try to move this so you can see the podium.
Hi, David. Great talk. Just a quick question.
Yep.
So for the fidelity calculations in your second experiment, did you do that only within like specific networks, or did you do that across the whole brain? Because I know you said something about like the part it's done through parcellation, right?
Yeah, exactly. So I think in this case, I'm presenting a Yao. I think this is a Yao-based parcellation. So it's the Yan, I believe, parcellation with like 500 nodes or something like that.
Okay.
We could do it in principle. We could do it voxel by voxel. I just did it with the 500 to sort of reduce the computational load.
That's fair.
Yep.
All right. Yep. Thanks.
Hey, David, I had just a couple of things. One clarification that I was just curious about from your first part. When you guys are doing your RSA and your leave one out, I'm familiar with the method, but haven't tried it. Do you remove outliers before you do? Or because there's so many subjects, does it kind of like wash out?
I did not remove outliers. So yeah, the thinking is because there's enough participants, it washes out, and it's tough to, I guess, say to define an outlier, because there's a
Yeah.
lot of different cells, for example. So when you just look if someone is pretty extreme within a cell, however, if someone just is very different from the rest of the group, I haven't removed outliers in that way. In principle, I don't think there's a reason why you shouldn't, but I have never done it.
Yeah, I guess my thought was when I was looking through was that if you maybe wouldn't want to, right? Because the idea is that some voxels will be particularly responsive to a thing, right? Like a face voxel in the FFA should be something that's activating a lot. So
Yeah.
as you were going through, that just got me curious, having not like done the steps before, what you guys' take on it was, but I think that makes sense.
Yeah, definitely.
And then I was hoping to ask more of like a concept question for the second part.
Mhm.
So I've worked with the gradCPT task a little bit, right? And I know for those like early papers, there's this idea of when you're like in the zone or out of the zone for like how your
Mhm.
attention is focusing. Do you think if you did this like second half of the analysis, but particularly focused on either those in or out of the zones, would it make a difference?
So well, I have done the zone-based analyses, though I haven't really coalesced them yet. So I have
Yeah.
images in a folder that I haven't really looked through, but there are certainly zone effects that exist, and there are these interactions with the zone. What I haven't looked at is the zone by valence effects. So splitting it up into like 4 as opposed to doing 2-way interaction, or I guess that'd be a 3-way interaction.
Yeah, just
I
I
think
wonder,
that
like, would my attention, like, when I'm out of the zone, is it because my attention is pushed more towards those scenes? And I think it'd be interesting to see, like, if the time course of in versus out of the zone is different across participants, or if it lines up, can it help us, like, better pull those signals apart when they're like in the zone versus out of the zone or something like that.
Yeah, absolutely. I mean, so I think it's funny because there's, I think, one old paper, at least older gradCPT paper, at least, that sort of posited there's sort of resource accounts essentially that say when you're in the zone, you actually have leftover resources. And so I think in the task, what they showed is sort of flanking scenes. And that they were actually, or maybe flanking faces. I can't remember. I think maybe the gradCPT was with faces and scenes were flanking. And they found greater priming effects of the scenes or neural habituation effects of the scenes when participants were in the zone. And so the idea being when you're in the zone, you actually have leftover resources to the periphery to pay attention to the background. So that
Oh,
finding would
interesting.
predict that you'd actually have more of
Yeah.
the background fidelity when you're in the zone than out of the zone. However, it's also possibly true that looking at the scenes will put you out of the zone, and that's sort of like lapsing your attention, and then you start
Yeah.
performing more variable. And so I think there's room for both to exist. And what you might find is that some areas of the brain have this sort of resource, processing capacity, and other parts of the brain, maybe the, slight timing of it is a little bit different, but other parts of the brain might be more sensitive to, the distraction. So they might be more responsible for switching your attention and getting you out of the zone.
Yeah, no, I wasn't familiar with the resources part paper. I just knew from like the behavioral stuff of the in versus out. So that's really, really interesting. Okay, any last thoughts or questions for David before we wrap up? Great. All right, thanks so much for your time. Really appreciate it.
Yeah, definitely. Thank you very much.
Bye.